Hello everyone, I am Debolina and welcome to my channel. In this channel, I teach biology to class 11th and 12th students and also to NEET aspirant as I am myself. So let's dive into our first chapter of class 11th, that is cell, the unit of life. What is a cell? Let's start with the basics. Cell is a structural and functional unit of all living organisms. Say it prokaryotes, say it eukaryotes, for both. We're gonna come to that on the later half of the chapter. So yeah, now we have Anton von Leeuwenhoek. He was the first one to see and describe a living cell. Second, we have Robert Brown, who discovered nucleus. Third, in the year 1838, we have Matthew Scriven. He was a German botanist who examined a large number of plants and discovered that all plants are composed of different cells and different tissues. Third gone. Fourth, we have Theodore Scriven, which is he was a British zoologist according to the old NCERT, and is a German zoologist according to the new NCERT. So go with the new NCERT. So Theodore Scriven in 1839. There's a year later. There is a year later, Matthew Scriven. He was a German zoologist who studied different types of animal cells and reported that cells have outer thin layer called the plasma membrane. Now, plasma membrane is present in both plants and animal cells, and cell wall is present only in plant cell, like cell wall, then plasma membrane, and then cytoplasm and nucleus. But in animal cells, we have only plasma membrane and then cytoplasm and nucleus. Now they both proposed, Scriven and Stirling together proposed a cell theory, which said that all living organisms are composed of cells and products of cells. But the question was, where do these products of cell come from? This question was solved by Rudolf Virchow in 1855. He was the first one to explain that products of cell come from the pre-existing cells. Like the pre-existing cells divide to form new cells. This was known as omnicellulari cellula. Done. Now an overview of cell. So now we go to the difference between prokaryotes and eukaryotes. Eukaryotes have a prominent nucleus and a prominent DNA inside the nucleus, whereas prokaryotes do not have any prominent nucleus. It doesn't have a prominent DNA. It has a naked DNA. Prokaryotes also do not have any cell organelles like endoplasmic reticulum, Golgi bodies. These are all absent in prokaryotes, but it's present in eukaryotes. The only organelle which is present in prokaryotes is the ribosomes. Ribosomes. What is the function of ribosome? The function of ribosome is that it does protein synthesis. Now ribosome. Where is it located? In plants, it is located inside the chloroplast. In animals, it is located not in animals, like in both plants and animals, they both have mitochondria. So inside the mitochondria, there is ribosome. And also in the rough ear. What is the meaning of rough ear? Ear means endoplasmic reticulum. So endoplasmic reticulum are of two types: smooth type and there's the rough type. Smooth means only endoplasmic reticulum is present. Our rough endoplasmic reticulum means that on the endoplasmic reticulum ribosome is present. So the location of ribosome also includes that. Okay, we have another cell membrane, non-membrane bound organelle, which is centrosome, which has a cell division. Now we come to the cell sizes. So there is a cell size varies very much from the smallest cell, that is of mycoplasmas, the smallest cell, which has a length of about 0.3 micrometer, 0.3 micrometer. Whereas we have the largest one of the ostrich, the egg of ostrich. You know that, right? Which is the largest isolated single cell. Human, our RBC length is diameter is about 7 micrometer, and nerve cell nerve cell is among some of the longest cells. Now, different shapes of cells include disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular. Now we come to prokaryote cell. So remember the points I've just told you. They do not have a nucleus. They do not. Have. They have They have naked DNA. They have naked DNA. These 2 points are you just need to remember. Examples of prokaryotes are bacteria, BGA, that is blue-green algae, mycoplasma. I just told you it is a size of 0.3 micrometer. And we have PPLO, pleuropneumonia-like organisms. These are the 4 examples of prokaryotes that is mentioned over here. Now, bacteria. We know the different shapes of bacteria. That is bacillus. We have rod-like. Coccus, we have spherical. Vibrio, which is comma-like, and spirillum, which is spiral shaped. Okay, so all prokaryotes have cell wall, except for mycoplasma. Mycoplasma do not have cell wall. It is an exception to prokaryotes. Have a smaller DNA called plasmids. They do not have organelles. I just told you that. Prokaryotes have something unique in form of they have inclusion bodies. A specialized differentiated form of cell membrane called mesosome is a characteristic of prokaryotes. They are essentially enfoldings of cell membrane. So mesosome is a unique character of prokaryotes. Now, for the prokaryotes, we have a cell envelope and its modification consists of 3 layers. We have the outermost glycolax layer, then we have a cell wall, and then we have the plasma membrane. Glycolax is a special characteristics of prokaryotes. Yeah, these 3 together becomes a protective unit, a single protective unit. Now, bacteria are of 2 types, like on the basis of how much they stain, or like do they stain or not. If they stain, they are gram positive. If they do not stain, they are gram negative. Now, glycolax, which is the outermost layer of prokaryotes, they are of 2 types. They can be, if they are loose sheath, it is known as slime layer. If they are tough and thick, it is known as capsule, tough and thick. Now, the outer one is done. Middle one, we have the cell wall. Cell wall is provides a strong structural support to prevent bacteria from bursting or collapsing. If you keep a prokaryotic cell in a concentrated medium, then there is a chance of bursting and collapsing, right? In just extreme weathers and conditions. So it just provides support from that. We come to mesosome. A special membranous structure is called mesosome, which is formed by extension of plasma membrane into the cell. These extensions are in the form of vessels, tubules, and lamellae. So, extension of plasma membrane is called mesosome, which is in the form of vessels, tubules, lamellae. They help in cell wall. Now, what is the function of mesosome? Cell wall formation, DNA replication, distribution of daughter cells. They also help in respiration, secretion process to increase surface area of plasma membrane and enzymatic content. Remember these points. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacteria cells may be motile or non-motile. By motile, it means that they can move from one place to another, or they can be non-motile. So motility depends on the presence of flagella. If there is flagella, then it will move. In absence of flagella, it is known as non-motile. Now, flagella is divided into three parts: hook, filament, hook, basal body. Filament, hook, basal body. The filament is the longest portion and extends from the cell surface to outside. Besides flagella, we have villi and fimbriae. Now, don't get confused. Don't get these two confused with flagella, because they do not have any role in motility, bacterial motility. Villi is elongated tubular structure made up of special protein, and fimbriae are small bristle-like fibers sprouting out of the cell. Small bristle-like fibers coming out from the cell, fimbriae. Villi, it is made up of special protein, and it is an elongated tubular structure. In some bacteria, they are known to help to attach bacteria to rocks in streams and also to the host tissues. Now, this line is very important. Like, it is NEET PYQ, and you need to remember this. Yeah, just for the NEET aspirants and also to the class 11 students, just mark it important. Now, we have ribosomes and inclusion bodies. The length of ribosome is about 15 nanometer to 20 nanometer. Now, let's just remember what I have just told you about the mycoplasm. Mycoplasm, the length was 0.3 micrometer, and bacteria it was 3 to 5 micrometer. Yes. Now, ribosome, we have the 50S and the 30S unit, which together form 70S, not 80S. Just don't need to add it up. When you will get to class 12, you will learn that from DNA, replication takes place to form DNA again, then transcription, then translation. From DNA forms mRNA by transcription, and mRNA forms protein by translation. So, DNA forms mRNA by transcription. Ribosomes get attached to mRNA and forms polyribosome. Like this, it is known as polysome. Just remember that. The ribosomes of polysome translate mRNA into protein. Now, these play a role, like mRNA plus ribosome, that is the polysome, play a role in translation. That is the formation of protein from mRNA. Now we come to inclusion bodies. Inclusion bodies are reserved material in prokaryotic cells. Reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. So it is like a storehouse for prokaryotes. These are not bound by any membrane. Like we have the membrane bound organelles. Here we have non-membrane bound. Non-membrane bound. And is free in cytoplasm. Example: phosphate granules, synophysin granules, and glycogen granules. Now we have a special gas vacuole which is found in blue-green algae and purple and green photosynthetic bacteria. This is very important. This part of the whole point of inclusion body is very important. Now finally we come to eukaryotic cell. Eukaryotic cell, let's remember the points I just told you. They have a prominent nucleus, they have DNA. Now, cell membrane is composed of lipids and proteins. Cell membrane is composed of lipids and proteins. Major lipids are phospholipids that are arranged in polar violet towards the other sides. Let me explain to you that. So, cell membrane is composed of lipids and proteins. Now, two lipids are phospholipids that are arranged in bilayer. Phospholipids meaning the meaning of phospholipid is phosphate plus lipid. Also, the lipid are arranged within the membrane with polar head towards. So, the structure of lipid, we have a hydrophilic head and a hydrophobic tail. So, the hydrophilic, what is the meaning of hydrophilic? Hydro means water, philic means lover. Hydro means water, phobic means you have like hate. Like we have phobia, right? So phobic, and philic means lover. Like we have in chemistry electrophile and nucleophile. So nucleus lover is nucleophile, and electron lover is electrophile. So like that, hydrophilic head. So hydrophilic head. Now, water loving. What is water? Well, like what type? Is it polar or non-polar? It is polar. Water is polar. So like hydrophilic can remain in aqueous medium, but hydrophobic tail cannot. This hydrophobic, these are the hydrophobic tails. They are non-polar. So so as to like protect it from the aqueous environment, they are inside. Like it is situated like this, and another is situated like this, and we have the bilayer formation. Bilayer formation. So as to protect from aqueous environment. In addition to phospholipid, membranes also contain cholesterol. Later biochemical investigation clearly revealed that the cell membranes also possesses proteins and carbohydrate. The ratio of protein and lipids varies considerably in different cell types. In human beings, the membrane of eukaryotic erythrocytes (RBC) has approximately 52% of proteins and 40% of lipids. So, the cell membrane is made up of lipids and proteins. Now, the amount, like the concentration of proteins and lipids, like varies considerably. But for RBC, we have 52% protein and 40% lipids. Now, the proteins are here. Proteins are of two types: integral protein and peripheral protein. Now, integral protein, by the name we know that integration, like joining of like the protein which joins two bilayer is called the integral protein. Like we have integral protein like this, and we have our bilayer over here and over here. This is known as integral protein. Then we have the peripheral protein, which is like moving, like it is like partially like it is lying on the surface and moving outside of this bilayer, whereas integral protein is partially buried or like inside, wholly inside the this bilayer thing. Next, an improved model of structure of cell membrane was proposed by Singer and Nicholas in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid structure of lipid enables lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. So Singer and Nicholas in 1972 proposed fluid mosaic model, according to which like it determines the fluidity of like it is due to the fluidity nature of lipids. The fluid nature of membrane is also important from the point of view of function, like cell growth, formation of intercellular secretion, endocytosis, cell membrane, etc., cell division, etc. Now we have passive transport. Many molecules can move, like molecules, the transport in which molecules can move from like one layer to another layer. This is a membrane. Like molecules can move from here to here, with here to here, without any like without any using of energy. Like it can move easily. This is known as passive transport. Whereas if energy is needed, like energy is required for this transport, it is called the active transport. That is the sodium potassium pump. So if the movement requires energy, it is energy dependent process. It is called active transport. Whereas it doesn't require any sort of energy, then it is passive transport. Now for osmosis we know that for water may also move across this membrane from higher concentration to lower concentration, which is known as osmosis. Now the water comes inside the cell, but the cell membrane which is like we just talked about, it is non-polar because it has the hydrophilic and hydrophobic tail, like both are present in the cell membrane. So if the cell membrane is non-polar, whereas the water is polar, how it is gonna come inside the cell? Carrier protein acts. Um, the carrier protein with the water molecule is gonna come inside the cell. Is gonna pass through the cell membrane and come inside the cell. Yeah. So let's keep it till here. I'm gonna post the explanation video of the second half of the chapter as a part 2 very soon. If you like my teaching and this video, do subscribe and like this video. And if you have any problems or any questions from this chapter or any chapter of class 11th and 12th biology, then do comment it down. And think of me as a friend who is just helping you out. And this video is also helping me to study because I'm an aspirant myself. So yeah. Thank you for watching. Keep studying, keep growing, and bye.